When we pour a glass of milk, the physical weight leads us to assume that a thicker pour correlates to higher nutritional density. But our intuition about how milk behaves as a fluid often contradicts how it functions as a biological fuel. To test this, we're comparing 4 specific samples: the commercial full cream, the commercial double-toned, the raw buffalo milk, and human breast milk. We'll evaluate these samples through 3 checkpoints: physical density, microscopic lipid structure, and a clinical lab breakdown. For new parents, this data is a matter of safety. Assuming animal milk replaces human milk based on appearance ignores an infant's specialized biological needs. Understanding the physics of milk reveals exactly which organism it was engineered to sustain. We begin with a lactometer test to establish fluid density. Pure water serves as our baseline, measuring at exactly 0. Because full cream milk contains the highest concentration of fat, the common hypothesis is that it will produce the heaviest, most dense reading. The data shows the opposite. Full cream measures at 32, while double-toned measures higher at 37. Fat molecules are less dense than water, so high fat yields lower density. Removing fat to create double-toned milk increases the heavier water ratio, driving fluid density higher. The raw buffalo milk sits between the process samples, yielding a reading of 36. Judging the quality of a milk by its thickness is a mistake. That physical sensation is a result of fluid physics, not caloric or nutritional concentration. But beyond the physical weight of the fluid, we need to look at the microscopic structure of the fats themselves. Under the microscope, raw buffalo milk is characterized by a high volume of mid-sized fat droplets. In contrast, double-toned milk shows lipids shattered into fragmented, sparse structures during industrial processing. The human breast milk sample stands out for the exceptional size of its fat droplets, which are significantly larger and more concentrated than any of the other samples. These large lipid droplets carry high concentrations of DHA and ARA, specific fatty acids providing raw materials to build infant brain cells and support cognitive development. The structure of the fats indicates the intended consumer: fragmented lipids for adult maintenance or massive droplets for rapid brain growth. Clinical lab results let us look past the visuals and measure specific protein loads in each sample. The data for buffalo milk reveals a massive protein concentration, far exceeding human milk. This protein is primarily casein, whereas human milk is dominated by whey. Casein is a heavy protein required for calves to stand within hours. Conversely, the clinical score for breast milk protein is just 0.9%, shockingly low next to the bovine data. While this low number might cause concern for parents, the specific protein deficit is a protective evolutionary feature. High casein animal milk presents a dangerous protein load that overworks an infant's underdeveloped kidneys, as their system is not yet equipped to filter such heavy concentrations. Adding more protein than a body can process is a physiological burden, not a nutritional benefit. Testing also tracks non-caloric markers that are essential for infant survival, specifically the concentration of iron. Animal milk lacks the bioavailable iron found in human milk. Substituting the two increases the risk of infant anemia, as animal milk cannot meet early development demands. We also found human milk oligosaccharides, or HMOs, which were unique to the breast milk sample. These complex carbohydrates are not for the baby to digest. Instead, they serve exclusively as fuel for beneficial gut bacteria, building the infant's immune system. We can map this clinical data into a decision tree. For adults, choose based on fat-to-water requirements, not physical thickness. For infants, animal milk must be avoided. The heavy protein strains kidneys, and the lack of HMOs and iron leaves their health vulnerable. Health depends on matching a milk's specific nutrient profile to the biological capacity of the consumer.